So hello everyone, hello everyone again, and welcome again to another Saturday live stream. Microbehunter here. You can also call me Oliver. Oh, I see I have to get the microphone cable a little bit out of the way here. Okay, here we go. I think I'm now all ready and set. Hello and welcome again, everyone. Today I want to talk about this little interesting microscope slide that I've got here, which I just recently obtained. Before I actually start with the live stream, I would like to, as always, ask you please to give me a short feedback concerning the sound, because I cannot hear myself. Sound is perfect. I can already see here. Great. Thank you very much. I mean, you can see that I've got a little, headset microphone here, which hopefully is also able to improve the sound a little bit. And I see that many people are already saying hello from all over the world. It's become a tradition, if you're new here, that you quickly introduce yourself, because microscopy is a hobby that is enjoyed all around the world. And what I will do today is I want to, as always, read out a little bit here from the people who are from where they are. Hello from Costa Rica, from the UK, okay, from California, okay, from northern Germany, 30 miles east of Hamburg, okay. So sound is perfect here. Where else? I'm interested to see what you have to say about the crystals. I have just had an exhibition at a London gallery of crystal photographers that I took with the help of my microscope. Yes, and I just, I don't know, maybe some of you just, was it yesterday or the day before yesterday? So also something that I wanted to say, there is it's called the Quebec Microscopical Society in the UK. They have had also just yesterday a full day of microscopy online Zoom video conference, and this was very interesting. I also joined that, not the full day, but I just wanted to say that there are also other possibilities to connect up with people here. Yeah, so a slight echo, but coming in clear. Interesting about the echo, because I completely turned off the sound that I have. Okay. Sound and video is great. Hello from Vienna. I'm doing some cross-polarized crystal photos right now, so perfect timing. Yep. And today I also want to show you a little bit some of the not so successful results that I've got here as well. From Ontario, Canada, and so on. You know what I'm going to do is I think I'm just going to get started here. So look what I got here. This is a little micro-aquarium that I got. I have to tell you, I don't know how much it was, because it was added I ordered something, and the company simply added it and gave it to me in addition. So it's not sponsored or anything, but I also do not know how much it actually costs. And it's quite an interesting little device. I've been experimenting today with the micro-aquarium, and there is also a sheet of instructions. It's written in German. But I'm going to translate a few parts here, because this is a really fascinating little device. It came along with a pipette, the standard pipette, but there is a little rubber cap here to make sure that it's airtight if you need that. And that's pretty much it. And how does this actually work? This is there are two little glass containers here, and if you add some liquid over here into one of those bottles here, then the liquid is going to spread. There's a little channel here. It's going to spread all across here into the other one until there's an equal level. And then you can put a cap on here. There's even a little Teflon thingy in here. And then it actually said here in the on the instruction sheet, that you are able to keep certain cultures up to 1 year. In this here, that's quite something. So this is, and the instruction manual also suggests a couple of interesting experiments that you can do with that. And indeed, one of the experiments I actually tried today, but I'm still working on it. I could not quite, it was not quite successful yet. And that is I want to study, and maybe I'm able to do that in one of the upcoming live streams. I want to study the reaction of Paramecia to electricity. So if you've got Paramecia in here, and if you put a wire into those containers here, and if you add a battery, it says, I've got like a 9 volt battery. I bought those as well. Then what the Paramecia are going to do is they're going to move towards the negative pole of the battery. And this is something that I would like to try out, and apparently you're able to kind of direct the paramecium around this way. It's, I think, quite a fascinating little experiment to determine this, and this is something that I would like to try. The instruction manual also suggests that you're able to add some live stains. So if you've got some organisms in here, then you can add some small amount of stain. You can watch the live stain, and you can add food, as it said, and you can actually observe it as well. Yeah, and the reason why this actually works so well is because, of course, there cannot be any evaporation here. Yeah. Because of the cap here, and there is a sufficient amount of liquid in there as well. Now, I think, however, that you should leave it open in any case, or at least occasionally, to allow for some oxygen exchange, because you certainly do not want this to become anaerobic, because in this case, those microbes are going to die, right? And what I would like to do first is I'm going to show you a little video that I made first, because if you've joined in the last couple of sessions, you've seen that not all of the demonstrations kind of worked. So what I've done is the following. I'm going to first show you a video, and then I'm going to add some sample here, and then we're going to have a look here. And the video looks like it's a Paramecia, because what happens is if you tilt the thing under the microscope, then there is, of course, the liquid is flowing in there in this kind of streaming, and I was able to see that the Paramecia tries to move. They try to move against the water flow. It's kind of funny. It's interesting to see. So here we go. Okay, that's the video now, and you can see that this paramecium over here, okay, this one over here, right? Yeah, is trying to, this one here as well. Yeah, they're trying to fight against the flow of the liquid. Yeah, and here is another one. Yeah. So it's quite interesting to actually see that those little cells are able to sense this. Yeah, and then it's starting again here. Okay, so you see that this one here, they're trying to flow against. Yeah. The water is streaming towards the right side, and it's trying to move towards the left. Yeah, these here as well. Yeah. So it's and what I would like to do is I would like to add some electrodes, and I would like to actually see how this actually works then with electricity. So these are the crystals here. No, that's not something I wanted to do first. So what I'm going to do now is I'm going to show you, before I do that, the instructions here. There is even some, it actually shows you how to fill it up here, how to use the pipette with a marker. However, there's one difficulty that you're going to see is because of the those containers here. It's a little bit difficult to place it under the microscope, so there's not enough movement possible, because some objectives might actually bump into those containers here. So it might be even useful maybe to remove some of the objectives, but it does work. Okay, but the movement is a little bit limited. Yeah, and there are a couple of examples here of how you certain experiments, and it actually says here over here it can euglena can actually be stored here up to 1 year. That's kind of interesting. Yeah. So that's something that I was kind of happy that I received this, and what I'm going to do now is I'm going to simply take a water sample, and with a couple of this here are some algae. And over there I have some, where is this? There's some rotifers in there, and some occasional Paramecia as well. And you were just gonna add it, and there won't be too many of them, but you just get the idea, right? So what I'm gonna do is I'm going to, you know what? I found out that this pipette, or I'm gonna show you here, is a little bit too small, okay, because it might get clogged. So in this case, what I suggest is that you simply take scissors and you cut off the tip a little bit, okay, to simply make it a little bit larger, so it's easier for you to take up some of the samples without actually becoming clogged here. Okay, so I go in here and I go all the way down here, and try to take up some of this material that is in here, and I put it in here as well. Okay, I tried this in the morning, and I have to tell you, it's a little bit a question of luck as well, of whether you're actually able to see a lot, because of course those cells are going to be spread over a larger area, and it's difficult to find them if you are not able to move it around very much. So another thing that I sometimes, or what I've tried is I've actually taken a little bit of solid material as well, and also, this one maybe not so much here, but some solid material as well that I added in here. Okay, simply to increase the concentration a little bit. Okay, and then you're supposed to fill it up a little bit more. Okay, and then if it doesn't move over, and it does not move over, then you're supposed to use the pipette, and maybe I'm able to apply a little bit of a vacuum here. Okay, yes, and there's still some air bubbles here. And yes, here we go. Okay, now I sucked it all the way through, so there is now a little bit here and also a little bit here, and then let's hope that we're able to see something. Okay, so usually there's always a demonstration effect, so I'm going to remove the crystals, which I'm going to show you later. It's always this demonstration effect that usually doesn't work so well when you want to show it to someone. That's why I made the video. Yeah. You can now see here at the top a little bit that there is a little bit of a problem here, because it's going to bump into the objective. For this reason, you have to move it all the way over here. And here we go. And I have to focus a little bit here. I'm going to move the condenser out of the way. I'm going to remove the polarized light here. Yep. And this is basically what I'm able to see. Yeah, and you see a small bunch of rotifers already. Okay, unfortunately, I cannot go up very much with the magnification because the objective is a little bit in the way. But maybe I can try it with a 10 times, and maybe I'm able to move it just enough. Here we go. Okay, and I'm therefore able to observe. Maybe I have to move out of the way a little bit so that you see it better. Yeah, and this is basically that I'm now able to observe some of the water life, and this is now, of course, a question of where are they. Here is another one down here. So so the movement is, of course, a little bit limited, but I'm going to add a little bit more liquid now, and this will now start the water flow again. So I have to go all the way up here. It's kind of difficult for me to do that. Yeah, and you can see that there is now a flow of the liquid as it starts to flow over. And so this is an interesting little device. And of course, the first thing that I thought of is this not somehow possible to make this yourself? Okay, so this would be the next project. Is to make maybe something similar yourself. I think I might have an idea. And one possibility, there was something kind of moving up there. Here, this here at the very top seems to be also a paramecium. Look at the very top here. I don't know why it's not moving. This one over here. Okay. So let's find it. Okay, yeah, I'm bumping a little bit into the objective again. Let's go down with the magnification because this gives us a wider field of view. And go down a little bit with the light intensity. And yes. So that's now the 4 times microscope objective. And you can see that there are, there's a rotifer over here. Yeah. So this is something I just wanted to show you as an interesting little addition here. Yeah, of course, something here, something over here. I don't know if you're able to see it quite well now on screen. Yeah. And if you tilt the whole thing, then of course this also causes the water to flow, and then it's going to flow back. And this is how I actually made the short video before. And then I had to zoom in a little bit so that you see the cells better. So I think this is an interesting thing, but I would like to now remove the liquid again, and I would like to show it now with some algae that I have here. Okay, so these are, that's a different specimen. So I have to remove everything that's in here. But before I do that, I've seen that some people have posted again a few comments here. So maybe I'm going to go in here Okay, so from Kentucky. Okay. Yeah, there's somebody. I was a speaker for an hour at the World Microscopy Day too. Yeah, so maybe it's 8 Thunder 8. So maybe I actually saw if you're the one who did the exhibition on the different crystals in polarized light. Yeah, I like this a lot. Yeah. Evening from Scotland. Okay, from New Jersey. Okay. I think the echo is simply from the room. This could be because I do have a room which is a little bit empty. From Michigan, Slavomir. Okay, that's very nice. Okay. Yeah, 1 year, Tienemansen. Yeah, that's at least what they claim. But I think, yeah. It is listed as 24 to 30 euros. Okay, I found the website of this. I'm going to show you this very quickly. Okay. So this is the website. I put the web address on the bottom here. Okay. And yeah, if there is even a possibility to kind of look at the picture here, but there is no price on here. Okay. 24 to 30 euros. Yeah, I mean, I wonder if there is something. I mean, I don't have to store the samples for up to a year, right? That's not necessary because I keep them in a jar, and that's where you can, and then if you want to look at it, I'd simply take a sample out. But one of the things that I kind of like here is because of this movement that's possible, so that you're able to actually see the direction of the movement of the microorganisms. And I would like to experiment a little bit around with electricity here. So, but the first thing that I would like to do now is I would like to again remove the liquid that is in here. And I simply do that by taking it out, and because I cut the tip, I'm not able to go in all the way. So for this reason, I have to, you know what I'm going to do? I'm going to try something different. You know, what I might do is the following. I'm going to simply, I've not tried this yet. I'm going to try some with water, and maybe this works if I flush it through with water. No, it didn't quite work. It does work. Yeah, it's now I spilled a lot of the water, but actually, yeah, there is now quite a bit. Yep, this does work. Okay, there's still a little bit of solid material in here. That's kind of hard to get out. I'm going to take it out maybe with some tweezers here. Okay. If not, then I just leave it in there. I'm going to exchange the tip because it's too short for my purposes. Yes, here we go. And I'm just going to flush out everything again. Yep, and I'm just going to let it run out simply naturally. Yep, okay, and I think now it's more or less clean. And I also tried today a few algae. So these were some rotifers that were in the sample, not too many of them. But over here, I got some algae in here, some single-celled algae, and they too. If you put them in here, okay, you wait until this kind of starts to flow to the other direct end. Okay, and let's have a look now. Let's remove all of the excess water. Okay. And let's see if we're a little bit lucky here. Yep, and that is at 4 times these are single, mostly of them are single-celled algae. But they are kind of small, so I think we do go up a little bit with the magnification now. Yeah. Then you're able to observe basically, what's going on here. I might be able to switch on DIC simply because it's a little bit more colorful, and you get the idea. Okay, let's move the arrow out of the way because it kind of disturbs a little bit. Yeah, and then you're able to, if you want to look at something else, you can actually tilt it a little bit. And then when you make it flat again, then everything's flowing back. Yeah? So I think this slide does offer a couple of interesting possibilities. Yeah, just started experimenting with this today. So yeah, there's not much that I can really show you otherwise. But I think there are a couple of yeah, experiments that I'm going to do in the future. Maybe some live staining might be a possibility. Because of the larger amount of liquid that you have, you can, yeah, the stain is diluted a little bit more. So there are several possibilities here. Just wanted to show this to you. So I'm going to go through again some of the quotes here, or the comments here. Okay. It was up to 1 year. The brand is Hecht. Yes, Hecht. Yeah, I'm buying this. Well, yeah, some people want to buy this. Yeah, I would say, why not? But I'm going to try to make something similar myself. I still don't know how I would do this with the small containers though, but I think if you use maybe some epoxy glue or some glue kind of maybe to glue some cover glasses, for example. I'm going to switch over now again to the scene desk. The thing that I was kind of thinking of, maybe somehow cutting some of those large cover glasses that I have here. Okay, maybe it's somehow possible to break them or to cut them, and then you're able to maybe, you know, using some epoxy glue or other glue maybe to kind of glue two of these next to each other and then maybe one on top. But how are you going to do that with the containers on the side? I don't know. Yeah. So another funny thing I just want to show you here. I was kind of wondering a little bit, what did they cross out here? Did they cross out something? And was able to just barely read it. It was patented. Yeah. So basically, I think the patent already expired. That's why they crossed it out. Yeah. But somebody actually invented this here and got it patented. Yeah. So I think that's something that one might actually try out here. Yeah. Yeah. Yeah, it's time to shock the microbes. Yeah, that's electricity. Some microbes actually do respond to not only magnetism but also electricity. Okay, you have to be the voltage, of course, has to be correct here. Yeah? Any chance of doing a soil microscopy video similar to your water one would help loads in agriculture? Yeah, as a matter of fact, this was a couple of years ago. Actually, a soil company contacted me, and because they needed some promotional videos, and they were actually making compost, different types of compost, and they requested simply for demonstration purposes and for showing to the customers some of the microorganisms that could be found in the different types of compost, and they didn't require any identification or any scientific study. They just wanted to have a couple of nice videos to show to the clients and to the customers. So they sent me a whole range of different compost samples. And what I have done is you simply mix the soil with a little bit of water. And then you basically put it on the microscope slide. But because sometimes the concentration is still too low, I made some enrichment cultures as well. Yeah. So could it be used to determine the aerodynamics of microbes? Yeah, rather than aquadynamics. Yeah, it was the movement of microorganisms in a stream of water is indeed something that can be observed. Okay. It will be interesting to see how the microbes respond to different voltage in real time with variable DC power supply. Yep, and that's indeed something I want to do. I have to tell you, I'm a little bit crazy maybe. But I've been thinking about something, but I don't know how to make that. It is known that certain micro, like Paramecia, they move towards the negative voltage pole. But if you could do some kind of a two-dimensional one, you know, you have a voltage on the x-axis and a voltage on the y-axis. And if you kind of control it with a joystick, you know, and apply the voltage like that, maybe you could actually remote control the Paramecia under the microscope. That is actually a project I wanted to do already, but I still have to think of how to actually get it set up like that. But if you've got any ideas, you need to basically have two ways of controlling the voltage, one on the x-axis, one on the y-axis, and then maybe with some joystick or so, you're able to actually remote control the Paramecia. Some crazy ideas that I had. Okay. Okay. Yeah. Hello from Spain. Okay. Yeah, where is this? Say you want to replace the culture, how would you clean it? So as a matter of fact, the instruction says the following. Yeah, replacing, where did I put it? It's still it's still on here. What they actually say is the following, is you should simply use this pipette to flush it. Yeah, so I might as well try that now. Okay, just like I tried before. You take the pipette, you remove all of the excess. Yeah, I'm just gonna save this again here. Okay. What I'm doing right now, of course, is I'm cross-contaminating it because in here there was the other water sample as well, but who cares? I'm aware of this. Yeah, and then you just fill it up. Yeah, and then basically what they said is the following: you're supposed to use this, and you're supposed to, yeah, press against it and kind of, yeah, suck it in and out. Yeah, I'm just gonna, yeah, and that's pretty much it. Yeah. And the instructions also say that it can be heat sterilized in steam. So if you have an how do you call this, yeah, a heat sterilizer, an autoclave, obviously forgot that name. If you've got an autoclave, which is a pressure cooker, then you can of course also sterilize this if this is necessary. Yeah. And because for me, I'm not doing any studies. I'm just doing some casual observation. It's not necessary to actually get it sterile. Yeah. So that's basically what they suggest. I would probably do the following. I would flush it properly, because if something dries out, some solid material, it might be difficult to remove. Okay. So for this reason, it's a good idea to really make sure that it's properly, properly cleaned before you actually put it away. Okay. Journey to the Microcosmos did a video on that. Yes, I know. Journey to the Microcosmos, the YouTube channel, actually did a video on the movement of Paramecia in an electrical field. Yeah, it's the closest thing to dancing paramecium I've seen, sort of like synchronized swimming. Yep. Okay. If you want to keep the sample alive for 1 year, how and how much food would you put into the sample? I would not overfeed it. And as a matter of fact, the instructions say that if you want to keep the sample, you should occasionally add a small amount of bacteria or food. But generally, there's always the problem of overfeeding. Yeah? Make sure that in any case, you should make sure, whatever sample you keep, to make sure that it remains aerobic. There should be enough oxygen contact. So putting the cap on it might prevent evaporation, but there's the danger that the whole thing becomes anaerobic, especially if there are no green algae in there that actually do photosynthesis, or cyanobacteria. Yeah? What does it say here? Yeah, you have to exchange 1 drop containing with bacteria with a pipette. Organisms like amoeba and various ciliates, rotifers, yeah, and algae can be combined as a community and can be kept in a micro-aquarium and observed. Okay, an easy. And here it says here, an easy experiment would be the reaction of single-celled organisms on electrical stimuli, galvanotaxis of Paramecia. Okay, and you have to basically put a wire through both containers and 3 to 10 volts, and then the organisms go to the minus pole. And if you reverse the polarity, then they're going to move into the other direction. Okay. And it can also be used to observe particles with a lens, magnifying glass. Yeah. So you see that it's quite an interesting little thing that I've not yet seen before, and that's why I kind of wanted to share it with you. Okay. So I quickly go through a couple of other comments here. Fun to watch. If you rotate the microaquarium 90 degrees, do you think that the bumping problem? Okay, that's something I didn't try yet. So let's do the following. Let's rotate this somehow 90 degrees. That might actually work. I have to. How am I going to do this? I have to put it behind. Nah, there are other objectives there. No, not working. Okay. Yeah. Somewhere over here. How? No, I cannot get it through here. Maybe. Yeah, and then you know what? Oh, it does work. Oh, that's cool. I just don't clip it in. I just place it on top here. Oh, thank you for the suggestion. Now it works. Look, I'm not clipping it in. I'm just yeah. But now I didn't. Oh, okay. That's actually quite nice. See, but there's nothing in it now. But this actually works. Yeah, you just place it on. You just place it on the clip. Yeah. I just cleaned it out, so there's nothing in here now. Yeah, so, and it's a little bit out of focus as well. Ah, there's still some. See, this is something that I could not get removed. Okay, that's okay. Thank you for the suggestion. That's actually quite a good solution here. I have to get my head out of the way so that you actually see how this looks like. Yeah, on top. Yeah. It also makes the container a little bit more easily accessible this way. Yeah, because it's more easily possible to add something. Oh, okay, that's nice. Okay, so something a bit unrelated. Does anyone have tips on how to observe daphnia, since you can't use a cover glass since you will crush them, and without it you can't use higher magnification? So what I suggest is the following with daphnia. I'm going to show you something that you might want to try out. The thing with water fleas generally is that they move around quite a bit, right? I don't have any water fleas here with me now, otherwise I would show it to you, unfortunately. And they're fairly large, maybe 1 millimeter, 2 millimeters in size. They move around very quickly. You want to limit their movement somehow. If you put a cover glass on top, you might end up crushing them to the extent that actually they get squashed. You don't want to do that. One thing that I highly recommend is that you do put it into a drop of water without cover glass, but that you use some tissue paper and you suck up most of the water so that the water drop is very small. And then this limits, because of the surface tension of the water, this limits the movement quite a bit. Okay, so this is suggestion number 1, and suggestion number 2, I'm going to show you as well, because it's a little bit related to what we've got here, is if you want to prevent them from being crushed, what you do is you take simply, okay, I have to open this now, it's a new box of, no, I've got an used here, and all you do is you simply make a bridge. You put a cover glass here. You can, if you want to, you can actually also glue it on with how do you call this, nail polish, but you don't have to. Yeah, and you stack 2 or 3 on top of each other, and you basically to keep a distance, and then you can place the third. And then I'm just going to use a large one across here. Yeah, so this allows you to keep a distance. So that is strategy number 1, to kind of lift up the cover glass. Strategy number 2 is to use a little bit of clay to make little feet on the cover glass. Okay. That's also a possibility. I've also used some nail polish to make a little feet here to kind of lift it up. So there are a variety of ways that you can experiment around to prevent it from actually crushing it. Yeah? So there's, but I would probably try this first. But for certain water fleas, lift it, just one cover glass might not lift it up high enough. Okay, so maybe, I don't know, maybe a piece of a credit card. I think that might be already a little bit too thick. Yeah. So, but that would be one possibility here. Okay. That was me. Ah, that was you. Okay. So this is good. 8 Thunder 8. I loved your presentation. Okay. So, it's still possible to see the presentation afterwards. Well, that's something that I cannot answer. Okay. I know that Quack at Microscopical Club do post videos of events that have been done. So I expect you could, but I think you have to be a member to see. Yeah. What I've seen is that if you want to join some of those Zoom videos that they're offering, and the reason why how I found out is because the link was posted on the Reddit on the Microscopy page on Reddit. Somebody posted this, and there was actually a full day program of all sorts of different interesting topics from the early morning until really late at night, and I was able to join in a little bit later, and I really liked the presentations. Yeah, and the reason why I like those presentations is because you feel that you're also part of a community, just like this here as well. Okay. That would be fantastic for taking high-speed photos in the movement of microbes. I think cleaning is one of the few drawbacks. Yes, that is indeed, yeah. When I'm going to inhibit the movement, well, not today, because I was not successful today. So the question is, am I going to inhibit the movement of microbes? Not inhibit it, but kind of move it around. I have to see if I'm going to be successful. I will tell you what the problem was, okay, with electricity. And I think that is the reason why in the video from Journey to the Microcosmos, when they have done that, they've used actually a power supply. And what I have tried to do is I tried to use a large 4.5 volt battery and also 9 volt battery. But I could not see any effect. And then I measured it out, and there was also no voltage drop. Now it's getting a little bit technical. And so I think that maybe I need to go up a little bit. I have to make sure that there's a certain amount of current going through it. So maybe the resistance was a little bit too high. So I need to see if I'm able to maybe obtain a power supply to actually try this out. Okay, so this is something that also Journey to the Microcosmos has done. They have used a power supply, not a battery. Okay, you may consider to try a concave type of glass slide. It might help facilitate your need. However, with the downside of the focus target due to the depth of field. So there are those concave glass slides which have an indentation. And they are also able to store a larger amount of water, and they are also very useful for thicker specimens like daphnia and water fleas. Okay. What? I've got a price, 268 pounds for the Microlife slide online. No, okay, 268 pounds. Honestly, look. You know why? No, this is not. Are you kidding me? Not 26.8 pounds, but 268 pounds? Wow. No, I got this slide. This Microlife slide was given. Basically, was given to me in a deal. It just said because there was I had a little bit of on a voucher. I still had a little bit of money. And instead of, so I asked the company, the supplies company, just give me anything. Yeah. And that's what he gave me, simply to kind of round up the value. Yeah. But 268 pounds? No. Okay. I'm a developer. Can I program? See, I would use an Arduino for the remote control project. Yup. That's something. Yeah. Hello from Switzerland. Sounds amazing when your microbial video game works. Yep. This would be actually kind of a fun thing, you know? You have maybe some kind of a maze, and then you have some kind of remote control, and you're kind of guiding the Paramecia through the maze or something like that. Okay. Where did you find that price? I found mostly in the 24 to 30 euros range. Okay. Okay. Moving Paramecia. Think about it. If I make it, I will tell you. Okay. Sorry for the light path is polarized or UV. So, and I will slowly start to move on a little bit to show you some of the crystals that I promised you. There's actually also a special cleaning solution available for this slide, the Microlife Cleaner. Okay, I didn't know that. That is interesting. Okay. You wouldn't need your X or Y axis at the same time, which would not be possible. You simply put the positive charge of one or the other in the four sides opposing the negative. Yeah, oh, that's the, okay. Yeah, so basically you're just rotating. That you only use two poles at the same time. Yeah, and then you kind of, when you want to move left or right, then you kind of just switch polarity. Okay. Okay. So it's for, okay. Sharper help for first time. What a great find. Okay. So what I'm going to do is lots of comments, but I would like to now move on a little bit to show you some of the an experiment that was less successful. I've been doing when I started, many years ago. I've been doing a lot of crystals, using especially vitamin C and so on. And today I wanted to try something new, and I was not successful, but I'm going to show it to you anyway. And I'm going to show you something where it actually did work out a little bit better. And just want to share this with you. What I've got here is the not so successful experiment of vitamin C. And what I've done here is I've actually placed a drop of saturated vitamin C solution on a microscope and then I added a cover glass, hoping that as the water slowly evaporates, it's going to make crystals. Okay, and we've seen indeed crystals did form, a lot of them on the side, but almost none of them inside. And I think what happened is that as the crystals formed on the outside, they've kind of drawn the water out, and the crystallization only happened on the outside. Okay, so what I've done therefore is I've placed some of the liquid here directly on the slide itself to allow it to evaporate directly. Okay, and this is the system that I've used. If you want to do a little bit of polarization work yourself, so I'm going to simply show you now what you can do, and this works with pretty much any microscope. Okay, this is and that's not the ideal solution, but it kind of works. There are those 3D glasses. They're called a Real D. They have the Real D standard, and they're circular polarizing glasses. However, if you can get linear polarizing filters, then they are to be preferred. Okay, and those circular polarizing filters, essentially, you cut them out, and then you have to find an orientation. It's kind of difficult to see. Where when you cross polarize them, that they become completely dark. And because they're circular polarizing, for this reason, there's only one orientation that works, and I think that's this one over here, right? And then when you put an optically active substance in between and light from the bottom, then it starts to shine in different colors. And so what you do is you take one of those filters and put it beneath the microscope slide or on your lamp. But it works better if you put the microscope slide directly on top of it, because sometimes if you don't have, sometimes there's a little bit of depolarization in the optics. And the other one you place on top of the slide. Now, if I've tried now of placing it over the eyepiece, shouldn't make a difference. No, it doesn't work over the eyepiece, and the reason is because microscopes that have a prism system inside to direct the light and to split it up into two eyepieces, these prisms, they depolarize the light. So you will actually lose the effect. So what you have to do is you basically use low magnification anyway. So you simply put something directly over it. In one of my videos, if you search this channel, I actually made a do-it-yourself solution with a rotating filter on top. Yeah. So I've got a couple of instructional videos on how to do that, and also some more in-depth videos on how to do crystallization. Okay. So basically, what I have done is I've put it under the microscope, and I've already got a polarization system because I've got a DIC. And I'm going to switch over. Okay. And when you put it on here, okay, I have to focus again. Then you're able to see the crystals that have formed. Okay. I'm going to go down a little bit. I can play around here with the colors as well. I will tell you now a couple of problems, or not I wouldn't say problems, but some difficulties here in making slide crystals. And I can actually start to make one here simply to demonstrate this. Okay. And then I'm going to, we're going to look at a few things here as well. And I'm going to now take a fresh tip, because I've prepared, actually I do have it already here. And I've prepared some saturated, vitamin C over here. And when you use vitamin C, where did I put the thing? Oh gosh, no, I bought myself, put it away. I don't know where I put it now. Don't use vitamin C tablets, but get yourself a pure crystalline vitamin C powder. It looks almost like sugar or salt. So it's not pressed in tablets, but it's pure. And you simply dissolve until, there are a couple of crystals not dissolving anymore. And what I have done in addition, I've added a few drops of alcohol to it to break the surface tension. And when you basically put it now on a slide and spread it apart, okay, what's going to happen is that the water will evaporate, also the alcohol will evaporate, and as it evaporates, crystals will start to form. The problem that I found is that if you do that, in some cases, the water will evaporate, but because of the surface tension, the drop will become smaller and more concentrated and will make a very thick layer of crystals. And sometimes they don't look quite as nice. So what you want to have a very thin layer. So what I found is that sometimes it depends also a little bit on how clean the slide is. Sometimes if the slide is very clean, then this has actually even a negative effect, because the dirt that you have on the slide, it acts sometimes like crystallization seeds, and it also prevents the surface tension from actually making the drop smaller. Okay, so then you wait a little bit, a couple of minutes, half an hour or so, and then you will see that like over here, it will start to turn quite white. Yeah, and depending on the concentration of the vitamin C, and depending on the temperature and also moisture and how fast crystallization happens, different types of crystals start to form. Yeah, and what I sometimes like to do here is or we can actually do it directly over here on the microscope here. This seems to be already completely dry. Yeah, what you can do is of course you can always add a little bit more of this solution. Yeah, and then basically see how it starts, how this starts to dissolve and then starts to recrystallize again. Yeah, and of course this one is already quite saturated, and for this reason, I've also made some beautiful images this way, and you can play around with the colors. You can rotate this. In my case, I just can rotate this here. And then you can also make some time-lapse videos. So we can, where's this slider? If I wait a little bit, then I'm actually able to see that the crystals will slowly start to form again. So we can try this again. Yeah, you see that I added some liquid over here, and the crystals, they did not dissolve. And the reason is because I already used the concentrated solution of vitamin C. So if you want to actually try it differently with to actually see them dissolve, then you simply add a little bit of water. So I'm going to add now some water. Okay, and the water should actually now dissolve the crystals. Okay, and then when the water evaporates, okay, you see this, the crystals start to become smaller. Yeah, you see those little that's because of a different refractive index. Yeah, see the crystals now starts to dissolve. Yeah, and then as the water evaporates, it starts to recrystallize. And if you play around like that, you can get quite a few beautiful patterns. Yeah. Again, if you're interested in this, then I recommend that you maybe watch some of the my previous video where I explained this a little bit in more detail on the whole things with the filters. And that's essentially if you are, that's one of the things that it's probably that would first things that of the improvements under quotation marks that I would try out is to play around with those with polarization filters, because they're easy to implement and the effect is quite beautiful. So I'm going to go over again to some of the questions here or comments here. If it was made of plastic, it would be expensive, but for glass, I mean, it's pretty challenging to produce something, and it's certainly not at a high volume compared to regular slides. Well, those slides you cannot make of plastic, because it's got to be, if you want to get a proper image quality, it's got to be completely smooth. Okay. Would you consider to use Wi-Fi or cell phone placed nearby the microbes to see if they will respond? Magnetic field from coil could be considered or experiment as well. Honestly, I don't think that, it's my personal view. Of course, they might respond to radiation and so on. But I think in order for you to be able to see something, this has to be quite directed, I would say. Yeah. But I don't know if this is actually, I mean, they wouldn't, let's put it this way. There are certain microorganisms that respond in magnetotactic, then they kind of respond to magnetism. So that is indeed possible, but just general radiation like a Wi-Fi or a cell phone, I don't think that this is even strong enough to actually cause, honestly, I don't know if they would be actually able to sense this, because what purpose would there be to pick up those types of frequencies? Yeah? I mean, yeah. Does anyone know what magnification human blood cells are visible? Yeah, I can tell you. You can start to see them. The red blood cells already with 100 times, 10 times objective, 10 times eyepiece, but they're quite small little dots. So if you want to see them properly, you need at least a total magnification of 400 times. Okay. The light path is polarized or UV. Are you asking? In my case here, this is this is not ultraviolet, of course. It's not fluorescent. This is polarized light. See, it's kind of dissolved now. Okay. Here's some more. Okay. Let's try to find this place where here. Basically, that's right at the corner. Okay. So we're gonna see. Maybe we're able to see how it starts to recrystallize after a couple of minutes. Okay. Applying plastic grommet stickers, laminate until thick enough to hold the sample, water, and cover. Okay, I don't know those stickers. Maybe double-sided tape. I don't know. Maybe some kind of a glue gun, heat glue, epoxy glue. I don't know. Yeah. Okay. Let's 100,000 subscribers. That's something I'm counting towards already. Okay, up here it is. Yeah. They are about 7 to 8 micrometers in diameter, the red blood cells. So 20 times objective or above will do it. Yeah, exactly. Yeah. Should the light in the microscope be always turned to its maximum power and access if it is limited just by aperture condenser ring? So, okay, I will tell you the following. That's an interesting technical question. And why not talk a little bit about technical issues as well? See, the crystals continue to start to dissolve. Okay. So, I want to show you something. This here, I'm now turning the light intensity. Okay. This is how light intensity should be controlled. When you open and close the condenser, of course, this also changes light intensity. But you should not use the condenser as a main way of changing light intensity. The condenser controls resolution and specifically depth of field and contrast. The change in intensity should only be seen as a side effect. You adjust the light intensity of the microscope depending on what is most appropriate for you. And at low magnification, you turn down the light. And at high magnifications, you have to turn the light up, because at high magnifications, there's generally less light. So you have to adjust it. So I cannot say that you should not always use it at maximum power. Okay. I will tell you, however, one thing: if you have microscopes that have a halogen light bulb, here light intensity is also important because it changes also the so-called the color temperature. So if you have a halogen light lamp, then if you turn up the light intensity, then the color becomes more blue. And if you turn down the light intensity, then the color becomes more red. So you have to use a white balance. You just have to know about that. So when when I'm taking pictures with a halogen, then I always use up the light intensity to the maximum because I want to get more blue light. Yeah. 268 for a case. They've given you a free sample, maybe. Okay, from Spain. In the high objectives, maybe 40 times objectives. Yeah, so this is still referring to the questions about the red blood cells. What liquids can make crystals? So if you want to make your own crystals, then I recommend, and especially if you want to try it out quickly and you do not want to wait to get some polarized light, you just want to make some very quick crystals. What you do is you just take a little bit of salt, make salt water, you make a concentrated salt water solution, and so basically you add a little bit of water, you add a little bit of salt, you make sure that it's properly dissolved, you take a drop of the salt water, you put it on a microscope slide, and you wait until it's dry, and then that's it, and you get beautiful salt crystals. They're not colorful, okay, because they're not changing the optical, they're not bifringent, or they're not optically active and so on, but it doesn't matter. You get very nice salt crystals this way as well. Yeah. So, but certain substances, like I've been experimenting also with citric acid. Citric acid gives also very beautiful crystals. Yeah. And I'm still, see, the water is still, because of the capillary action, still starts to spread. and is not yet starting to dry. No, on the other side here. Yeah, so I'm gonna wait. Yeah, I can do some. I've made some beautiful time-lapse videos as well. Yeah. That's really funny. I'm photographing vitamin C crystals right now. Yes, that's a coincidence. Yes, please do share your experiments as well. I have to tell you that the crystals that I have here again look quite different to the previous ones that I've made. So it depends quite a bit on the conditions that you have. Okay. How did you get a vitamin C from a pill? No, don't get it from a pill, because when you buy vitamin C pills or those vitamin capsules, they contain not pure vitamin C, but a lot of other stuff as well, filling material. So, you know what, I'm going to show you, but I have to jump up. Okay, I'll be back in 10 seconds. So I'm gonna put this on again. So I'm just gonna show you what I bought this in the drugstore. It's pure vitamin C. You got to be a little careful with this stuff because you don't know how much you're actually taking in. Yeah. I guess it's also used for conservation, preservation of it's an antioxidant and so on. And the vitamin C looks like this. It is just pure powder, and why not? I'm just gonna show you what I've done is I've taken a little bit of water, and I added a little bit of vitamin C. Okay, until I even warmed it up a little bit in the microwave until no more vitamin C could be dissolved. Yeah. This one is not going to dissolve now because it's already a saturated solution. So that's what I've done. So it's really easy in that sense, right? But if you want to quickly try out crystallization yourself, then I recommend that you try it out with salt. Okay. So seeing microscope here again. Okay. So I hope that they're slowly starting to crystallize. This takes some time. It also depends quite a bit on the moisture and humidity that in your area. So that's also very important because otherwise it might actually take quite long. Yeah? Change of light that makes the color. Is it polarized or UV? I'm using polarized light. UV light is used for fluorescent microscopy, which I don't have. Okay, so this is polarized light, and you can make polarized light quite easily yourself by passing the light through a filter like this here. Okay. I once bought a big bottle with powdered vitamin C from a drugstore. It was years ago, not very expensive either, and citric acid is very cheap as well. Citric acid you can get from even from regular in regular shops, supermarkets, larger supermarkets, because it's used not only for cleaning but also for making certain foods. Okay, so actually was hoping that this whole thing is going to start to crystallize much faster. That's again one of those demonstration effects. Okay. What should I do to guarantee that I can see exact, original, actual, natural specimens colors? I have noticed that the color depends on aperture setting, filter used, and I never know which one is correct. Okay. The aperture should not influence the color so much. What you do is you just go regular bright field. Okay. Regular bright field will show you the colors in regular light. Okay, and because those vitamin C crystals on their own do not have a color, they're white, right? You can create the color using the polarized light because the different wavelengths interfere differently. They extinguish each other, and then certain colors remain. If you want to speed up the crystallization, you can dissolve it in 99% isopropanol instead, and then turn on the crystals with a day, and it will be very thin. And we can, as a matter of fact, I don't have isopropanol now, but I have regular alcohol, and why not try that? You know what? I'm going to try it here. I've got some. I'm running out of containers now. Just a second. Where do I have a container? I'm just going to use this here. Okay. So I'm going to now use regular ethanol, okay, and this is ethyl alcohol, and vitamin C does not dissolve well in alcohol. Okay, so this is also something I just want to make clear. Normally, okay, then, but so. What I've also done is to maybe make it easier to dissolve, to add maybe a drop of water. Okay. So let's just try it out. Yeah, this might, it's slowly dissolving, but again, the solubility is not as quite as good in. So I'm going to, where is this here? So sometimes if you take some of the non-dissolved crystals, they also look kind of nice. Okay, so but let's try this. I'm gonna add and you see, because the surface tension is much less, therefore, and you know what, I'm going to add a little bit of some of the crystals as well, simply that we have some crystallization seeds here, and it will spread over the whole slide because of the low surface tension. And then let's have a look. You know what, we can have a look directly here right now, and let's put in the polarization again. And I have to switch over to the microscope view, more light. Yeah, these are still some undissolved. Look, some dirt, dust, whatever. And usually sometimes the crystallization starts at the edges where the layer is thinnest. But we can actually wait a little bit. And another thing that is sometimes quite fun to do, this was also demonstrated in the presentation of from the Quackhead Microscopical Society, was essentially you would use some of these cotton swabs, and then when you actually go across it, maybe I'm going to and you rub around it, then sometimes this will initiate the forming of more crystals because the crystallization seeds are started this way. Okay, looks quite nice. So, but let's be patient here a little bit. This is another thing that I just want to show you. Maybe you're able to see this here or not. Because of the low surface tension of the alcohol, some alcohol spilled over now, and this is beneath the slide. Okay, so it's simply also something that we have to then clean the stage later on. Yeah. So there's a little bit the thing here. So let's move it. Let's go on somewhat lower magnification here. Okay, let's wait a little bit. Maybe this goes a little bit faster. Yeah. So I'll read a couple of comments here. Yeah, I have videos, definitely. Yes, I've. If you check the channel, I've got actually more detailed videos. Okay. I could try to freeze water. That's going to be a little more challenging. Okay. I put water in the slide and then freeze it while the slide is under the microscope. That is, a little bit difficult, I think. Okay. There is one thing that I also would like to, I cannot demonstrate this now, but something that I would like to also recommend, and I also made a video on this. And what you do is the following. You take, it's a little dangerous here for me to do that. That's why I'm not going to do that here. But what you do is you take a, maybe you can see in the corner now, or let's just switch over here. You put a match, the head of an unburned match on the slide, and you take another match and you light that match, the match on the slide with the other match. So what you have is you have a quick fire of the head of the match burning, and you immediately blow it out. And then what you have is because of the phosphorus and the sulfur that evaporates, started to sublimate and recrystallize on the cold microscope slide. So you basically simply take that microscope slide then and put it under the microscope, and then you can see the crystals that were formed from the burning of the match head. Okay, if you search on my channels, I think I call it fire under the microscope or something like that. And then you can actually also, that's a very interesting way also to make those crystals visible. They're quite small, however, but plentiful. Yeah. So that's essentially a possibility of it's called sublimation, when you have a gas which then starts to solidify as it condenses on the microscope slide. Okay, something that I would also like to. Yeah. So how far are we here? There is something happening. Yeah, as it evaporates. Or I'm going to check the other one over here. That's the problem, because the slide got moist on the bottom side. I have to kind of dry it, because I spilled over some of the alcohol. Therefore, the slide does not move anymore. Look, it sticks to the slide, to the stage. Up. Yeah. So I have to clean it, and I have to clean this here again, make this a little bit dry, so that. So let's have a look now again. Yeah, here it is. Yeah, and let's see if the other one. Yep. What has happened is over here the alcohol already has formed some droplets here where it is evaporated, because unfortunately the liquid got spilled over. Okay, so that's again a demonstration effect here. But let's wait until they kind of become a little bit larger. Okay, so what time is it? It's 1:06 already. Okay. Yeah, our P can form crystals as well. Yes, of course they can. And I also made some videos here. Halogen light emits some light in the invisible ultraviolet spectrum below 400 nanometers, which is very damaging to human eyes. Does microscope bulbs have a filter which cuts the wavelength and the frequency below 400 nanometers? Well, as a matter of fact, when you have a halogen lamp installed, they often have you often use a blue filter, as a matter of fact, to limit the red parts of the spectrum, because the blue parts of the spectrum actually make the image a little bit sharper and give it a more natural color. Okay. I would say, I mean, people have been using halogen bulbs now in microscopy and also for many years now. Yeah, so I would say that it might. So there is no filter used, but I can imagine that maybe the glass also filters some parts away. Yeah, I've found that some plant foods make good crystals. They have a potassium nitrates and such. When we press a button or run app on the cell phone, the high-speed signal will be transmitted and received between the tower and the phone. The energy has to be high enough to do so. Yeah, but the question is it high enough to actually have an effect on the microorganisms? And is it going to be detected by them? That's the question. I'm not saying it is or it's not. Yeah. So I'm just saying that microorganisms have to be, the energy has to be high enough for them to be able to detect it. Microscopy, in a sense, the energy might be high enough to trigger the response. That will be my hypothesis. We can try that. Yeah. But the question is are we able to shoot the signal in a certain direction that they are actually able to get a directional stimulus? Yeah? I have a science experiment kit that is used for kids, and in there is a little container with citric acid. Maybe I can use that. Yeah, so what you can do, what I have done is to make citric acid crystals, but this takes a long time to crystallize. is to actually melt them over a hot plate very quickly and then put a cover glass on top. Then you can make those melts as well. Yeah. What is the concentration of salt and water for crystallization? What you do is you just add as much salt to the water until no more salt dissolves. So this means when you basically have salt crystals on the bottom of your jar, of your cup, and when they do not dissolve anymore, then you know that you have a saturated solution and then you can use that. If it is not saturated and you still use it, then as the water evaporates, it will become saturated and then it will crystallize. So just add enough salt that it does not all of the salt is able to dissolve. Worth pointing out that many crystal types can be made by melting rather than dissolving. Like, for example, I've done this with citric acid, but you got to be careful. If you heat it too much, I've also destroyed some of them. Yeah, so this basically means that because they're organic substances, if they're too hot, then they start to break down. I've tried making salt crystal slide, but it failed because it needed polarization. Honestly, the thing is salt crystals, if you use polarized light, you will not get any colors. Okay. So the reason is because substances that are polarizing, they have an asymmetry. They have often organic substances with a carbon atom that is which is asymmetrical, and therefore it changes the direction of the light. Salt doesn't do that. And for this reason, even if you use polarized light, you're not going to see anything different with salt. Okay. Electric coffee cup heater. Yeah, that would be something. So, and as a matter of fact, there are heatable slides as well. And honestly, this takes a long time again here because I think this has to do a little bit with the humidity in my room that I have. Yeah. The cotton swab was with saccharin. I've never actually tried it with salt. Okay, that's also an interesting thing. Okay, I can imagine that if you dip the cotton swab maybe in very fine salt grains, then these salt grains could actually be used. Maybe they are. Maybe these salt grains are then the crystallization seeds. Yeah, and what I've also tried to do once, and this I don't know if this works here very well. I'm going to try this now. Okay, is the following: is simply to use your finger and to carefully go over it. It's still very, very moist. Yeah. Usually it starts to become thicker and it starts to become a little bit syrupy. And then when you do that and when you streak over it, then you see there where you streaked over it, crystallization to start. And it's really interesting over here. It doesn't seem to want to evaporate. Yeah, it's kind of interesting. Okay. But you know what I'm going to do, just because now I'm getting a little bit, how do you say, excited about this, because I would like to know why. So I'm going to do the following. I'm going to try this again here a little bit. Yeah. On here. And I'm going to use much less water. But it could be that the whole thing is hygroscopic. This means that it likes to actually absorb the water from the air around it. This could also be a case. Okay. Now, these crystals are quite small here. So let's see the cotton swab. You see now a little bit the liquid here as well being absorbed. Let me see if there's something over here. Here there is still a little bit of liquid visible here. So let's see if crystallization goes on over here. Yeah, reinforcement label, circular gommet to modify slides, making a well inside the slides, similar to what circles you were painting before. Okay. Potassium permanganate also produces nice crystals. That's a very purplish, pinkish, substance. It really works much better with citric acid or salt in isopropyl. Interesting. Okay. Can you make your own citric acid from scratch? If yes, what is it made of? Citric acid is citric acid. I mean, of course, you could try to extract it somehow, but I think that's crystallizing it out of a natural source, like from lemons and so on. I think is very, very complex. Yeah, I think it's synthetically made. Yeah, and it's really cheap. Lighter fluid sometimes works, and you can even see the liquid boil away until it's extremely saturated, but very dangerous. Yep. A wave plate, a lambda 4th wave plate, can also add some colors. Look a lot nicer with polarizer. What are those crystals that are covering the whole slide? Those crystals that are covering the whole slide is vitamin C as well. I spread it across the whole slide. Okay. Maybe you can use lemon juice and then add some stuff. The thing is I think if you add natural, I think it might not work with lemon juice, because crystallization really requires very pure substances. And if you have a natural lemon juice, then there are lots of impurities in there that might actually prevent crystallization from happening properly, I think. So, and you know what? I'm going to now still try some of this here. I'm going to let it now evaporate a little bit here on my table to see actually what's going on. So, yeah, what else do I have here? Okay, so I'm going to wait a little bit longer and we're going to see how this works. Honestly, I consider this again fascinating, because this one here, yeah, seems to be, I show this to you. I added a very small drop of liquid, and there seems to be now more liquid here than there was before. So I actually do assume that it has to do something with the humidity in my room, that actually it doesn't dry quite the way that I expected it to dry. So this is a little bit, also an interesting thing here to see. Yeah. Let me check a little bit. Maybe I can show you some of the. Let me quickly check. Just a second. I have to go online. I will now check something because I want to show you maybe some of the pictures that I made before. Let me see. Yeah, here we go. I'm gonna show you a website. It's my online gallery, but just a second, I have to share this somehow with you first. How do I share this? I have to, just a second. I'm going to go here again. And I'm going to share this. Give me a second, please. Give me a second. Maybe I'm able to share it. I have to change the link here. Yes, here it works now. Okay, I have to click interact. So this is a website that I have here. Allow all. And here I've. Does this even work? How can I cannot move down here, unfortunately. Okay. Pictures and videos, non-living crystals. Is it loading or is it not loading? I think it's easy. Yeah. Some people also, here are some crystals as well. Yeah, this one, for example, this one is citric acid that I made some time ago. Okay, it gives those nice little flat crystals here, and I used a citric acid melt for this. Over here, this one over here is also citric acid. The thing is yeah, this one here as well. The thing is these pictures are quite old. I actually used analog film and slide film to take those pictures, and later on I digitized them. Yeah, but it just shows you. And this one over here is vitamin C, so it looks very different again. And it depends quite a bit on the way that the crystallization actually happens. Yeah, there's also vitamin C. Vitamin C looks very different. Yeah, this again is citric acid. Yeah, again, citric acid. It's actually one of my favorites. It's vitamin C. It looks a little bit like a comet. Yeah, it's also citric acid. This one is vitamin C. Yeah, it's described anyway. Yeah, yeah. Like yeah. Yeah. So I just yeah, and all of these things were done with my yeah, simply using some cheaper polarizing filters. That is basically from urine in oblique illumination. Yeah. I can pause here. They look a little bit three-dimensional, and this is because I had light coming from the side, and this kind of gives it a little bit this 3D appearance. And yeah, so this is simply a drop of urine on a slide, and then you simply allowed it to evaporate. Yeah. Yeah, so it's yeah, quite interesting. Yeah. So this oblique illumination is also something that I can recommend. You simply put a dark field patch stop under the microscope, and then you kind of decentralize it and decenter it, and then the light strikes the image from one side and gives you a little bit of feeling of depth. Okay, so I just have seen again that there are some comments here. Are there any crystals that don't dissolve when water is added? If you want to use aspirin, acetyl salicylic acid, then you need alcohol and caffeine. Yeah, I think caffeine also dissolves better in alcohol. Yeah, so it depends really a lot on how hydrophilic or hydrophobic they are. Yeah? What is the most likely place to have diatoms? Yeah, in water. What you do is you go out into a pond or a stream and you scratch some of the surface material off, and then you can find some diatoms there. But you know what? I've got. I can show you what you can try as well. Yeah. Or for example, when you have some algae floating on the surface of a pond, okay, then you take some of this filamentous algae, and then in many cases there's some diatoms in there as well. Okay. Okay. I want some diamond. Well, there are certain synthetic diamonds that you can also buy. Okay. Vitamin C and citric acid look very different, but assume that I'm much lower magnification. So I used at least, yeah. I used for most of the crystals, I used around 4 times or 10 times objective. Okay. Okay, so just some comments. There's some discussion. I'm just reading the comments here. There's some discussion going on in the community. What crystals are you looking at? Okay, right now vitamin C, citric acid, and salt. Okay, so yeah, there was a question about not related to crystals, but related to diatoms. Now, I don't think that there are a lot of diatoms in here, but what you do is when you get yourself a what time is it now? It's 1:22 already, so I have to slowly think about stopping anyway. If you want to get yourself a water sample and you see some green stuff floating, yeah, like something like this, on a pond, then what you do is the following: you just grab some of this stuff. Okay, here you grab some of this. You take a small sample. Use my scissors here, and then you do the following. Simply tap it, and you add a little bit of water as well. So I'm gonna exchange the tip again here. Okay. And what you do is this. Okay. There has been once somebody emailed me and asked me, well, what do you do? All of the organisms, they move around so much. There won't be a lot of organisms in here. I know that already, but maybe I'm able to get some single-celled algae out here. I say is don't use a lot of water, and to make sure that the cover glass is not lifted away by thick particles. So, and let's just leave it like this, and let's hope for the best. And you add a cover glass, and if you're lucky, you're able to find some diatoms in here. You can try the same thing with moss. You can try the same thing with by scratching some material from the surface of rocks. Okay, well, there is lots of stuff here already. Yeah, I don't know if there are a lot of diatoms or not. We're gonna see. It doesn't appear to be. I do have another sample over there as well. And if you want to see the whole thing in natural colors, this is how it looks like, and then you're able to see actually the green color of the algae much better. There might be some few tiny diatoms in there. Okay. These are. This is a that okay. The movement is so fast that you can't see this. Most of them are not diatoms. Yeah, but you get the idea. So you have to make sure that you're somehow able to get out the individual cells. So this here is most likely a diatom. Just a second, where's my arrow? This one over here. Okay. Those long ones. Okay. Okay, so can you tell me where's the gallery? What you do is the following. Yeah, this website is has not been super active now for quite some time, but I called it microworldarchive.org. Okay, just type it in here and you find the gallery on there, and you can also download some of the magazines that are published many years ago. Microworldarchive.org, and some people, it's not all of the pictures are my pictures. Okay. I put it into the, I send it now into the chat. Okay. That is, you can have a look at the gallery there. I posted a couple of pictures that I made over the years. Yeah. Not only vitamin C, but also some other things, and with a description. And a couple of years ago, people sent me also microscopy articles, and I made a magazine, and you can also download it there or from the web page. Yeah. So very quickly, because it's already over 1.5 hours, why is it that most, if not all, children's microscopes only have 3 objectives? Okay, I can tell you that. The thing is the following: children's microscopes, cost is of course one reason. Children's microscopes have a 4 times, a 10 times, and a 40 times objective. And the 100 times oil immersion objective is missing, and it's a good idea that it's missing, because the 100 times oil immersion objective is difficult to use and requires immersion oil. And if it's not properly used, you can actually damage your microscope. And so cost reason is one thing, and also because you do not need so much high magnification of 100 times oil immersion objective, and this would also require much more light. And in order to get the image quality, you also need a proper condenser. All of these things essentially push up the price, and for this reason, children microscopes have to be cheap, portable, and for this reason, they are kind of saving on certain places. Yeah. Yeah. Okay. Wow, that earthworm cross-section image is fantastic. So that is in the gallery, in the link that I posted. Okay. Yeah, and I think maybe that's a good idea to. Honestly, I've after I've watched the Zoom meeting from the Quackett Microscopical Society, I got myself so motivated again to take a couple of nice gallery pictures, so that I think that I'm also going to keep on updating. Yeah. Keep on updating this. Many of the pictures that I've posted there, I have of course stitched together. This basically means I've made overlapping images and put them together. Yeah. So yeah, and some of the images that I've published there on the gallery are from commercial microscope slides. Yeah. Yeah, so I think that's I think I'm going to keep them updated. I think now I have again a good motivation myself to post a few pictures there. And you know what? Yeah, the 100 times oil immersion, that's the question was, again, for children microscopes, the 100 times oil immersion objective takes a lot more work than dry objectives, and it is also a little bit dangerous, because what can happen is that if you put immersion oil on the slide and then the kids rotate the other objectives into the oil, and this is going to happen, then you end up actually damaging the other objectives as well, and you don't want that. Yeah, and yeah. And you need a proper condenser as well, and for this reason, I would generally say that many things that can be observed with children can be observed also, especially in low magnification. Yeah, so, and for this reason, it's also not necessary. Yeah. People, there has been a question. What's next week's theme? Honestly, I don't know yet. Really, so I have to admit, I'm a little bit improvising sometimes as well. And it depends also a little bit, the direction of a live stream depends also a little bit on the comments that I get. If you have any specific requests or so, then yeah, please do post them. I want to try out the whole thing with electricity, however, but I'm not so sure of if I'm going to be successful or not. Okay, can you make a bright field microscope to work like with Köhler illumination? Köhler, yeah, I mean, some microscopes, bright field microscopes do have Köhler. So yeah, the Köhler illumination is a diaphragm on the bottom that allows you to limit the diameter of the lamp. Yeah. Okay, dancing microbes. No promise here, but I'll try to get it done. Okay. First upgrade, I'm going to get my first is a 60 times. I highly recommend 60 times. Okay. 1 hour, 30 minutes is the time for me to stop. Okay. And water bears and keep them in the new slide. That's actually an idea. Okay. That's actually an idea. I've got plenty of moss. If you want to observe, by the way, I want to give you a tip, because some people have some problems finding water bears, and of course, they can be found in moss, but when you collect a moss sample, make sure that you get the moss sample from an area that did not get too hot. So I had a very big success in finding a lot of water bears in moss that are found in a forest. Because if the moss is outside, directly exposed to the sunlight, and completely dries up, and then there is also heat there, tardigrades, water bears are dry resistant quite a bit, but they're not so heat resistant. Okay, and in a forest, even if the moss is dry, then they have a highest chance of surviving. Yeah, and for this reason, if you want to collect your own water bears, try to get the moss from an area where the heat was not able to build up so much. Okay. Yeah, interactive species ID and count on the water sample could be good fun. By the way, a species ID just today in the main channel, I uploaded a video where I'm just showing you very basically a couple of species. Okay, for people as a little identification help. Yeah, yeah. Also, if the moss is dry, it's also necessary to add water and wait for a day or so. Yes, that is correct. You have to wait. When it's dry moss, you have to add water. You have to wait for a day or two. But even then I found that the moss that I've taken from a forest contained way more water bears than the moss that I collected from growing on the concrete in front of my apartment. We have a concrete wall there, and there was some moss growing there. And I collected moss samples there for the tardigrades, but I could not find quite as many water bears as the one in the forest. And I think, and that's my hypothesis, is because there's also the sun shining there, and it can actually get quite hot on the concrete. And the moss comes back to life again when it's raining and so on. But it could be, I think, that this heat might kill off the water bears, I guess. Yeah. Can you zoom in to look at those round diatoms in DIC? Okay, yeah, DIC. I need more light. DIC is very light intensive. I have to change this here. Yeah, I'm going up to 60 times. Change this here. Here a little bit more light, and yeah, I don't know. You choose the color. Again, those round things, these are not diatoms, right? The long ones might be diatoms here. Like the one here. This is most likely one here. And an empty diatom shell right next to it. I'm going to point this. Okay, so this is most likely a diatom. This appears to be like an empty diatom shell. Okay. If you want blue, you do the following. You experiment with something like this. Okay, it's a dark field patch stop, but not completely black. I 3D printed it. You can use blue nail polish to make something like this. Okay, this also works. Yeah. So this is by the way, if you really want to make it blue, there is lots of color adjustment that you can do in a video editing program. Yeah. And usually I use blue because or blue green because it kind of imitates a little bit the water. Yeah. Yeah, and back to 40 times. I have to change again the thing here. Yeah. Yeah, so that's basically. There was something moving here. Oh, whatever. Okay. Yeah, I think I'm gonna leave it at that, folks. Yeah, hope you liked it. And now I think I'm also gonna start to look a little bit more into maybe making a few more pictures with using some vitamin C. Yeah, if you really wanna see some nice pictures, I've never participated because there's this the company Nikon publishes every year a nice calendar. It's the small, I think they call it the Small World calendar, where it is a photo competition. They have extremely beautiful pictures in there as well. Okay, very, very nice pictures as well. Okay, I'm going to quickly read through the remaining comments and then I'm really going to stop. Okay, what are those round organisms? These are single-celled algae. Okay, unicellular algae. Okay, if it's green, it's most likely photosynthetic. Okay, thank you for having joined here. Okay, and there is they're in the form from the homepage. Then you can actually there are some people also posting pictures there as well. But I'm going to leave it at that now. Okay, I wish you all the best. I hope that you liked it, and you can have a look at the gallery, of course, if you want to see a couple of pictures there. And I'll see what I'll be able to prepare for next week. Okay, all the best. Have a nice weekend still, and happy microbe hunting as always. Bye bye.